Welcome all to this wonderful day, which is a holy day. Happy Holi to everyone. We have an esteemed guest, my good friend Sunil Polipalli, who is a senior scientist at Loknaik Hospital, Maulana Azad Medical College based in Delhi, where he is a renowned cytogeneticist. And he has been instrumental in coming up for various scientific breakthroughs in identifying various elements associated with newborn screening. His group and lab has developed some kind of projects that are associated with newborn screening, molecular cytogenetics, most importantly, prenatal screening, and also in lieu of epigenetics. But over the last 2 years, apart from the wonderful cytogenetics that Sunil has been taking care, his group and his lab has been instrumental in identifying unique variants and unique mutations with respect to Omicron variants of SARS-CoV-2. Their group have sequenced as many as, I would say, 500 whole genome sequencing of SARS-CoV-2 as well. So, moving back to his career achievements, Sunil has obtained his PhD from Department of Medicine, Maulana Azad Medical College, affiliated University of Delhi Faculty of Medical Sciences, in 2010. And ever since he has been working at this Loknaik Hospital in this COVID surveillance and as well as Department of Cytogenetics. And he has been instrumental in bringing a lot of collaborations with IGIB and various departments of cytogenetics at various places. And he's also the president of Geneticist Clinician Consortium of India, which is largely from Bioclues and as well as bioservices. So with this little introduction, I think we all should welcome Sunil, where we can learn a lot of other things. Thank you so much, Sunil, for obliging to my request on this holiday. Thank you so much, for your time. You may start your presentation, please.
Good evening, everyone. Thank you, Prash, for inviting me to have a conversation with you. So, I'm very happy to be part of Bioclues, Bioservices, and GCCI. So, actually, I'm involved in cytogenetics part as well as molecular and biochemical genetics at Loknaik Hospital. So, today I'm going to little bit explain exactly we are doing on newborn screening at our hospital. Uh, my team members are Dr. Seema Kapoor, Dr. Somesh, uh, other few members are also there. So, but main things we'll be working with Dr. Seema Kapoor, and Somesh is one of our PhD student. He has completed his PhD and he is also part of our team working closely with newborn screening. So, I'm going to explain a little about the newborn screening.
Yes. Sunil, are you sharing your screen, please? Yeah.
Yeah, yeah, I'm sure.
Okay. Thank you. Thank you.
My slides are visible.
Yes, yes, Sunil. Yeah, please. Yeah.
This is what exactly we are planning. I am going to explain. This is about what we are looking after the comprehensive newborn screening. Here you can see the three symbols. One is the heel, one is heart, and one is ear. So these are the three components what we are exactly looking into. This is what exactly every child deserves a healthy start in life. So it promotes a medical paradigm shift. It can be a model for genomic medicine, predictive, preventive, and personalized. So this program can be used as a
Excuse
public.
me, sorry to interrupt you. Your slides are not
Yeah.
moving. Yours is not in full screen mode.
Okay.
You can click full screen mode and yeah.
Now it's coming.
No, Sunil. But the slides are moving. The good news is, yeah, I think the slides are moving, but maybe you can refresh the page. Sometimes it helps. Or maybe this is also okay if you can move from one slide to the other like
Yeah,
this.
yeah. So
Can you go to the second slide now so that we can see?
this is the core elements what we exactly want to look. We can look up the birth defects,
Sunil, yours not,
So
yours is,
maybe you can, uh,
share.
no, yours is not in second slide, Sunil. Maybe you can click second slide with your mouse click.
so maybe you can just stop once the
Stop
screen
sharing.
share, and first
Yeah,
you can just go for entire screen sharing. You can do that, might
Okay.
be.
yeah, now it works. Yeah, this is fine.
Yes.
Yeah.
Now it is coming.
Uh, no, yeah, now, no.
So you can share your entire screen, sir,
Okay.
and then you can click on the PPT or the presentation that you want to present, sir.
Is it coming now?
No, Sunil. Yeah.
No, sir.
Sunil, you can directly click that share option and even earlier, it worked. I don't know why it's not. Yeah, now it's working. Yeah,
Now it's coming.
now it's working.
Yes, sir.
You can directly click the what do you call your mouse
Yeah.
slide so that you can. Yeah,
Okay.
thank
Okay.
you. Yeah.
Sorry for the interruption. Uh,
Right.
so regarding newborn screening, every child deserves a healthy start in life. So because actually nobody knows what happens to a baby when they were born. So what disorders might be there, nobody knows that. So in order to identify that, we can screen a baby, newborn baby. So it can be helpful for the genomic medicine, predictive, preventive, and personalized. And you can save babies 1 in 300 lives by biochemical testing, hearing, pulse oximeter screening. So the core elements of newborn screening, what we can look into the birth defects. You can see the birth defects. What are the defects that are visible when you see the baby? And metabolic disorders like congenital hypothyroidism and congenital adrenal hyperplasia. These are the metabolic disorders that can be manageable and treatable at the initial stage. And like visual defects, retinopathy of prematurity. The eye vision also can be detected at the early stage, and hearing defects can be rectified, and congenital heart disease also can be cured when identified at the earlier stage. And hemoglobinopathies can also be screened by uh, dead blood spot only, and these are thalassemia and sickle cell variants can be identified. And confirmation can be done by biochemical and molecular diagnosis. So why you want to screen a baby? Because uh, every baby requires a healthy life from any other genetic disorders like that. So in order to screen a baby, you have to collect the sample at 24 to 48 hours of birth, so that it can be collected on a dried blood spot. So if we identify at early stage, that can save the baby from any damage, early damage, and the mortality can be reduced. So how a baby is screened? So within 24 to 74 hours of birth, 2 to 3 drops of blood are collected from the heel of the baby on a filter paper for the tests like biochemical parameters and hemoglobinopathies can be done. And these facilities should be provided to the concerned hospital so that they can do the screening of the babies. This is what the how to collect the sample from the newborn baby. These are the line what has been given. You can collect the sample from here, these both sides. This is the actual red blood spot where to collect the sample of the newborn baby. So this SS, this part you can collect the sample. So these are the procedure how to collect the sample. And these are
Are the slides moving? I am seeing only that newborn screening slide.
okay. Slides are
The
not moving.
second, no, only the second slide. I think you are on second slide. Is it the case with everybody?
So
Yeah, I think Sunil, can you please click this, slide? Yeah, exactly. This is how you can click. Yeah,
okay.
yeah,
These are the samples how to collect the sample from the baby. So these are the filter paper. You can collect the sample from the one drop of sample. You should not over put two drops at the same position. Only one drop is required in the circle where it has been provided. This is how the sample should be collected. And this is for the birth defects, how to identify the birth defects of a baby by seeing only. So these are the guidelines of NHM to identify visual birth defects like left palate, everything you can identify. And this is the workflow what our lab has developed. So whenever the baby is screened, the collection coordinator, staff nurse, and the clinicians are integrated with the mobile app, and the sample have been collected, and it will be transported to the concerned lab. And the babies, for if they're having any congenital heart disease, they will be screened by pulse oximeter, and then necessary procedures will be performed by clinicians. And hearing aids also are also one of the parameter, hearing screening for deafness program, that also can be done at the initial stage only. These are the main thing, the workflow, how the things are being coordinated. So this is what exactly the program is. So how to screen a baby for genetic disorders which are treatable, and untreatable disorders also can be identified. So this is the program which is being implemented worldwide, but in India we are in a stage, in the primary stage only. So we are doing the primary newborn screening now in only few states. So in Delhi we are starting. We have started now recently, but this we have done for last 8 years. We are doing in a project mode, but now Delhi government has approved as a new mission, and we are doing these things now. And after this, once the baby has been detected with any abnormal result, then we call the baby for follow up, and we do the routine testing for other biochemical parameters and molecular testing. So this is about the primary newborn screening, what we are doing, and mostly we are looking after the thyroid and congenital adrenal hyperplasia. That is, in that 17-OHP enzyme is elevated. So based upon that, we can look after the how the enzyme is elevated, everything we can see. And after that, we can go for uh, second option. This is the expanded newborn screening, where you can do the sampling, everything. Already primary screening you have done. In that, you can see what are the metabolites that have been elevated or not, and if they are elevated, then you can go for secondary metabolite screening by GCMS and LCMS, primary newborn screening, and GCMS you will be going for secondary metabolites, and after that you can go for molecular analysis. If the baby is having some like PKU, yeah, homocystinuria, then you can call the parents and you can take the sample and you can go for counseling to them, and you can explain what is the disorder your child is having, and you can give parental counseling. So for next pregnancy, if they want to go for, they can be explained what are the consequences they are going to have. So this is what exactly we are planning to do the things. So thank you.
Excellent, excellent, Sunil. Yeah. So Sunil, moving further, to start with, how did cytogenetics, come to the fold of Sunil's interests?
Rani here. Hello, sir. Can you hear me?
Yeah, yeah.
Yeah. Indrani, please. This is Sciverstion, so which means, it's a kind of an interview kind of thing. You are welcome to,
Yes, sir.
yeah, welcome to ask questions,
Sure, sir.
after the
Sure.
Sciversation is over. This is not a webinar, please. Thank you so much. Please wait. Appreciate your patience.
Yes, sir. Thank you. Thank you, sir. So, sir, in the newborn screening, like apart from all the diagnostic tests that you do, I wanted to know is like
Sunil,
have been.
Indrani, please.
Yes, sir.
This is what I've been requesting you, please. You will definitely be asked, you'll be given an opportunity to ask questions. Please, this is a Sciversation. This is only a very preliminary thing. There will be a Q&A session, and then you can ask questions.
Okay, sir. Okay, sir.
I apologize for this. This is Sciversation.
No.
This is not
Yes,
a
sir.
webinar. Please understand that in a Sciversation, we discuss about what you call a conversational entities with respect to scientific ethics. These Sciversations are really meant to bring some kind of an inspiration to the young people, so that we ask the guests to start how come they are interested in their own fields, what aspired them or what transpired them to bring science to the fore. And then during the last 10 minutes, there will be a kind of a Q&A session. Okay, please. Thank you. I'll definitely get back to you on this. Thank you so much. So sorry for this. Otherwise, thank you so much for joining. So Sunil, thank you so much. We will definitely come, with a Q&A session. So Sunil, what transpired Sunil, to take up cytogenetics, especially when there are very few cytogenetics geneticists. I think at the helm of your MSc and your PhD, way back in 2010 when you finished it. What made your interest in cytogenetics?
Uh, Actually, I did my master's in human genetics from Andhra University in 2003. And my professor M. Satyanarayana sir and Dr. V. Lakshmi Kalpana, they have motivated me to do some cytogenetics part in spontaneous abortus. So I did after my masters. I 1 year I trained in Andhra University Human Genetics Department. I did a project on spontaneous abortus, cytogenetic abnormalities in spontaneous abortus. So my madam has motivated me a lot to do some cytogenetics part. So likewise, I came to Delhi in 2004 and I did my joined as a PhD student from Maulana Azad Medical College under supervision of Dr. Premashish Kar, co-supervisor from Jamia Millia University, Dr. Syed Hussain sir. So and Dr. Anil Agarwal from GB Pant and Dr. Ranjana Gondal from GB Pant. So under their mentorship and supervisorship, I did my PhD from Maulana Azad Medical College. So
Wonderful.
during my PhD, I worked on pancreatic cancer, on DNA methylation and P16 gene in chronic pancreatitis and pancreatic cancer. So after that, I joined in 2009 as cytogenetist in Loknaik Hospital. So from that time, I'm looking after cytogenetics part as well as molecular and biochemical part.
Yeah, wonderful, wonderful, Sunil. I mean, moving further, Sunil, at the helm of, doing your probably PhD when you call yourself as cytogeneticist, you are also doing, a major, piggybacking, working on pancreatic cancer, pancreatitis, and as well as, something, more related to newborn screening, getting, towards, karyotyping and all those, kind of things. So you have a wonderful, mixed bag of all these kind of things. But my question is, something different.
Yeah.
When you worked on pancreatitis or pancreatic cancer, there's a lot of insulin resistance mediated diabetes signaling happening in pancreas,
Yeah.
which means a lot of differential express genes might be upregulating at that point of time. Uh, what is that particular missing link when we try to observe a patient with say diabetes and a patient with pancreatitis, pancreatic cancer?
Yeah. Yeah. We looked cases with diabetes as well as non-diabetes also, and most of the pancreatic cancer patients are diabetics only, diabetics. So and it has been head region of pancreas. Most of the cases have been head region of pancreas because pancreas is having three parts: body, tail, and head. So most of the pancreatic cancer arises in the head region, and it is very difficult to identify in the early stage. So most of the pancreatic cancers are advanced stage. So it is not in the earlier stage, earlier ages. It is mostly in the advanced age group only. The with diabetes only. Most of the cases are diabetes cases, and I looked into the only KRAS gene and P16 gene. So mutation patterns of codon 12 and 13 in KRAS gene and P16 gene in P16 DNA methylation we have looked into.
Wonderful, yeah, wonderful, Sunil. Yeah. Sunil, moving further, I mean, you have extensively worked on newborn screening. But if you really see, my point is largely towards what we call it as Helsinki guidelines, which are largely towards the ethics, ethical guidelines that are promulgated by many scientists and many countries. What does Helsinki guidelines, say about the newborn screening, methodology, especially
Oh,
in India? Are we really following these
yeah.
Helsinki
Actually,
guidelines?
actually, for newborn screening, Wilson and Jugner criteria is followed.
Okay, yeah,
Helsinki rule also and Wilson and Jugner criteria. Based upon that, the newborn screening pattern is followed worldwide. So in India also we follow the same guidelines.
right. And could you please, give a small gist about these guidelines? What do they say?
In that, it is a universal criteria that you have to screen all the babies who are being affected, who to be screened. All the babies who are born should be screened for identifying any particular disorder.
Right,
So in that criteria, they are having around 29 amino acid disorders as well as 30 other parameters also that are not non-treatable. So like G6PD, thyroid, 17-OHP, Biden-Days, galactosemia. These are treatable and manageable disorders. But other things like PKU, homocystinuria, tyrosinemia, these are uh, treatable with the diet, and enzyme replacement therapy can be applicable. These are the criteria that are being followed worldwide.
wonderful, wonderful, Sunil. Thank you for this. Yeah. So moving further, Sunil, you have been, instrumental in publishing a lot on, you know, the newborn screening, the cytogenetic, mediated diseases, and all those kind of things. Can you probably pick one such particular paper, that is very close to your heart, and then, say that yes, this has been, a paper, of my particular thought, where I can, where, others can also take inspiration from.
One of my article is cytogenetic abnormalities, that is for 5 year study from our center. So in that we have screened all the cytogenetics part of Down syndrome cases, primary amenorrhea, secondary amenorrhea, IVF cases,
Right.
and we have framed guidelines for newborn screening. Likewise, this is for congenital adrenal hyperplasia cases. We have made a guidelines so how to screen the baby and how to go to approach. That guidelines have been framed.
Right. Wonderful. Yeah, moving further, slightly a different topic. I know, Sunil, very few would probably have done a PhD from a medical college, that, from a science background. Uh, how easy was it for you, or how difficult was it, for you, to cope up, some kind of, things, for a scientist, working or doing a kind of a PhD in a medical college, or how easy was it, from a learning point of view? Could you please highlight?
Yeah, initially everybody will be having some little bit hiccups, but later on we have to adjust and you have to go ahead, because starting stage, when I came to Delhi in 2004, I'm not having any fellowship, but my mentor has provided me fellowship in a clinical trial on hepatitis B. So I worked in a clinical trial with one of my colleague. Uh, he's Dr. Mohammed Asim. So now he's working in Dubai, and
Wonderful.
one of my colleague is also friend is there, Dr. Kausar Niaz. He also worked with me in a clinical trial, Himalaya drug. He worked on hepatitis B. So we did the clinical trials, and then we have little bit for our livelihood. Also, we have to earn something. We have to keep. So a little bit struggle, but later on, once we got registered for PhD, and we got fellowship from, we have written a project. Actually, I wrote a project on my topic and submitted to UGC from my guide. So after that, the project has come. So with that project, I joined as a project fellow. After that, senior research fellow in a clinical trial from ICMR on hepatitis B in cirrhosis patients. In that, we have given drugs, edifovir, lamivudine, glizazine in a clinical trial to all the excuse me, to all the hepatitis B positive patients. So likewise, I'm doing my PhD as well as a clinical trial also. Used to collect the samples, and it is easy for us to get the samples. That is the main advantage
Right,
when we are working in medical college.
right, right. And how easy was it, to get this informed consent, especially for newborn screening and, for the
Uh,
downstream validation and other stuff?
no, for newborn screening, we have to explain to the parents clearly what exactly we are doing about the sampling. So what, because they don't understand what exactly we are collecting the sample, because the newborn baby after 24 hours, so it is very difficult little bit. But now uh, all the people have been trained and we are explaining how to go ahead with the parents. So because we are saying that these parts are being implemented in India, and Delhi is the first thing that is being doing this program. So we have to explain to the parents what exactly, like thyroid, because thyroid they know, parents know, most of the parents know about thyroid, but other parameters they don't know. But we have to explain to them, खून की जांच हो रहे हैं, like that we have to explain. आपका खून का कमी है, तो बच्चा को खून देना पड़ेगा. Like that we have to explain. Like thalassemia,
Right,
hemoglobinopathies, and sickle cell, we have to explain to the parents. So likewise, we have to take consent. After that, we will be taking consent from the parents.
right, right. Wonderful, wonderful, Sunil. And this particular process, I mean, be it, from the receiving end of such particular process, be it as a wonderful scientist, in your group, were there any kind of disagreements that you have had with any of your colleagues, Sunil? So that, I mean, did you any time, convert those disagreements in an agreeable way? I mean, this is again a normal question that I usually ask everyone, so that, everyone, should learn the art of, putting certain disagreements in an agreeable way.
No, no,
Could you
I will
please?
say directly if there is any problem in the lab, I will say directly to my colleagues who are working with me. I will say directly.
Right,
Uh,
right. And were there any instances, taking on a wrong side? I know, that happens, many a time as well. But how did you put that thing in an agreeable way, and how did the other opponent receive it, or maybe you put it in a different way? Is
no,
there
no,
any?
no. I don't. I keep my cool and I go very coolly, and I will explain the things very coolly. So, if you want to work in a lab, any workplace, you should maintain some repo and you should go work together. If you want good results, you have to work together. That is the main motto what I follow.
Right. And I have a very subtle question, because you are a psychogeneticist, and you have extensively worked on something related to newborn disorders and all those things, and of course, cancer as a part of your PhD, you take it as a part of your postdoc and your job and other things. Uh, uh, I mean, if you really look into the newborn screening area, we are still in some kind of infancy when compared to the Western countries.
Yeah,
We might have, major screening maybe happening only in maybe in Mumbai, or maybe in Delhi, or maybe in Hyderabad, or in Bangalore, not across, all the states.
yeah,
Do you see that the newly established clinics or the tertiary care centers, that are being established should have such, newborn screening facilities? What is your take on this, Sunil?
yeah. Actually, now the government of India is making some policies to implement this newborn screening. Actually, now Kerala is having one program on newborn screening. Delhi is having, but DBT has started a program like Nidan Kendra, Umeed program. So they have started newborn screening for all the centers. What they have given funding, like 7 centers are being now doing the newborn screening all over India. Now this year, I think they may expand that part now by providing more infrastructure and facilities for newborn screening.
Right.
We are working for RIMS Ranchi also. We are getting samples of newborn screening from Ranchi, and other centers like AIMS is doing from Rishikesh. They are getting samples, and SGPJ Lucknow is having another sample from UP some place I don't remember, and CMC Vellore is also doing some newborn screening from their part, and CDFD Hyderabad is also doing some part. So now the newborn screening is coming into the proper picture now. So actually, this is the primary newborn screening what we are doing. Actually, now we have to go expanded newborn screening, because primary newborn screening you can identify only particular some disorders, but they may be false positive also sometimes. So
Yeah.
for that reason, you have to go for secondary newborn screening. In primary school newborn screening, you will be going for LCMS and capillary ectophoresis parameters. But for secondary newborn screening, expanded newborn screening, you have to go for GCMS and molecular part. That is what Western people are doing now for second time newborn screening. That is what.
Right. I mean, having said this, there is a very crucial need for taking the artificial intelligence forward, Sunil. I believe that, let's assume that, we have a lot of, newborn screening done. We have some kind of a metadata existing,
Yeah,
for all these particular things. And keeping in view of such future newborn cases, that needs to be analyzed in the future, can we apply some kind of, machine learning heuristics, or can we apply artificial intelligence based things, where we can say that yes, these particular phenotypes are something, similar to the already analyzed or already existing metadata from the previous cases, and then, we might, reduce the number of tests, for the newborn screening. Is this going to be a kind of a reality in the future, Sunil? Where are
yeah,
we
yeah.
as of,
It is,
as of?
it is, it is there. It is there. Artificial intelligence can be applicable because visual birth defects or images you are having. So
Right.
by seeing the images, you can integrate the database. And for the parameters like biochemical parameters, you can integrate the uh, predictive value, false predictive and negative predictive value, and you can set up a reference range of the samples, and
Right, right, yeah,
you can integrate your data
right.
likewise.
And how difficult is this, and what are the inherent challenges or probably limitations for this, Sunil, if you can kindly highlight?
No, we have to look into this because we have to have a huge data, so that then only you can have a positive predictive value and negative predictive value, and specificity and sensitivity of the testing. So likewise, you have to integrate all these things completely
Right,
with the health informatics system. Then it will be easy for you to maintain the database, and you can correlate your metadata with
right,
artificial intelligence.
right. Wonderful, wonderful, Sunil. I mean, just moving further, as a president of Geneticist Clinician Consortium of India, what would be the grand challenge, for the current state of the art of development, especially when we look into the GCCI as a kind of thing. We need, of course, I know, once we have started this consortia, we need to bridge, a lot of gap between clinicians and geneticists. Uh, what is the future like, for
Yeah,
GCCI?
because we can start, because genetic counseling is the main bridge between the clinicians and the scientists, because genetic counseling is not available to all the people. So we have to start some coursework, yeah, like short-term coursework to the MSc students, and we can provide training, because most of the clinics, genetic clinics and hospitals, that doesn't have a proper genetic counselor. And
Right,
if you get you are the better person, because you are the bioinformaticians, bioinformation, because when we get a sequencing report or microarray report, so how to interpret the result, because the clinicians mostly, it is difficult for them to understand the reports. So if because MSc students, they know bioinformatics, and they learn a lot during their course. So if we can provide some basic coursework idea about how to go for genetic counseling, then it will be very good for the students as well as for the upcoming genetic counselors.
wonderful, wonderful, Sunil. I mean, something on these lines, Sunil. I know genetic counseling is certainly, in need of that. But how does that really value, for any, consortia needs? I just will try to give you a kind of an example. As a systems geneticist, or as a systems biologist, or whatever, as a bioinformaticist, in fact, our job here is, to identify the bona fide variants, the accurate variants, whether or not, if we are given a particular data, we double check it, we triple check it probably, and then only, confirm whether or not a variant is pathogenic or not. But what's really happening with, even with the newborn screening, or even with the congenital cases, most of the time is the kind of, pipelines that people employ. They're not able to identify the bona fide variants. Some of the things, they wouldn't probably look into whether or not there are any autosomal dominance or autosomal recessive conditions, or whether or not, heterozygosity is seen in a large instead of, the homozygosity. All these particular, guidelines, including the Sherlock criteria, all these particular criteria are certainly given a miss. And I think, sorry, I think, GCCI, as per our white paper article, we are trying to do that. But what do you see as a scientist, as a senior scientist, in your lab, when you get, some kind of a report, say a whole exome sequencing report? Would you simply trust that particular report as is and say that yes, this particular report is, say, coming from some X company, and we simply trust that, this report is bona fide or true? Would you again, cross check it with some bioinformaticists if you get it out?
Yeah, we cross check with the other bioinformatics also, because most of the cases we get VUS only. So that's why we cross check
Right,
the,
right.
we get the raw data, and we used to check it,
Right.
reconfirm it.
For those, some of you who don't know what is VUS or VOUS or GUS,
uh,
genes of unknown significance or
Yes,
variants of
with the
unknown
unknown
significance.
significance.
These are all very important, is uh, which is, a kind of a predicament I would say these days, if we really get those particular variants, that's very unfortunate because they do not really map to any of those particular known existing databases, and that could really kind of a concern for many scientists. Even the genetic counselor has to say, cytogeneticist like Sunil has to say it, or maybe someone like systems geneticist I have to say, and we have to say to the parent as well, to look into whether or not the pedigree information is,
Yeah.
there or not. Unfortunately, these particular things are not really happening, and I think that's where probably we could probably reach that particular gap. Wonderful, wonderful, Sunil. I think I just have probably one or two questions which I'll probably uh, uh, ask you later, but I'll open this particular questions for the audience in the next 15 minutes. I think Indrani has a question. Sorry, Indrani. Yep, now you may go ahead, please. Please unmute your mic.
Yes, sir. Yes,
Yeah.
sir. That's fine, sir. Thank you, sir. So, sir, my doubt was like apart from the like the ultrasonography, that is the conventional technique that we have been using for decades to diagnose like a child's health condition and like the morphological appearances. Apart from that, any other like like technical tools which helps to identify any abnormalities in the fetus?
Yeah, you can go for amniotic fluid and CVS testing also. If your uh, parents are carriers for thalassemia, then you can go for CVS tissue extraction of DNA, and you can see the mutation. What mutation is there in the CVS tissue, so that you can explain to the parents whether it is a carrier baby, whether the baby is affected or not, homozygous, your wild type. So if it is homozygous mutation, then the baby is affected. Then you can say the
Yes.
baby is having mutation. So then you will be explaining about the complications what they will be facing. Because for thalassemia, the baby is homozygous affected, then they have to undergo blood transfusion continuously. So we
Yes.
will explain to the babies. And one more
Oh,
thing, for aneuploidies, we are saying ultrasound, na? Then if
yes,
you
sir.
for some parameters like Down syndrome, 21 plus, no? 21, trisomy 21, and 13, 18, and 21 aneuploidies. For that, you are having biochemical parameters like nipple marker, beta ECG, alpha fetoprotein levels will be checked, the biochemical parameter. If they are elevated, then you can go for this amniotic fluid culture. So in that you can identify whether the baby is having extra chromosome or not, aneuploid is there, that you can
Okay,
identify
sir.
the defect.
So these are all the like invasive techniques, like
NIPT,
under the non-invasive techniques. Any
non-invasive is NIPT. You can go for NIPT, non-invasive physical screening. In that you can take the blood sample of mother, and they will be doing self-repeated DNA extraction, and they will be looking for the aneuploidy detection. NIPS is one
okay,
of
sir.
the
Okay, sir.
techniques you can go.
Okay, sir. Okay, sir. Thank you, sir. It was very informative talk, sir. Yeah. Thank you. Thank you, Prashant, sir.
Yeah, wonderful, Indrani. Thank you. Thank you for
Thank you,
asking a
sir.
great question. So, any other questions, please? Please unmute your microphone and please feel free to ask Sunil. It's a wonderful opportunity. Yeah. As the questions roll in, I have two more very scintillating questions. Over the last 2.5 years or so, Sunil, I mean, your lab has been very much instrumental in identifying or sequencing as many as SARS-CoV-2, genomes. Uh, uh, how did this happen? And as from government of Delhi, I think it must be super excited for you, to sequence as many these genomes and helpful in lateral diagnosis as well. Can you please highlight about this sequencing facility that you have?
Actually, this is the initiative of Delhi government through our medical director, Dr. Suresh Kumar sir. Uh, it has been initiated. So Dr. Arvind Kejriwal, CM of Delhi, has inaugurated the sequencing facility, and this is the first genome sequencing lab from New Delhi, and the second one is ILBS. So we got all the samples from Delhi airport who have been travelers. So we received all the samples from December onwards. We got all the samples they have been traveled, because during the peak time that Omicron started in the December. So we have identified the first Omicron case from Delhi, from our lab. Uh, on December 5th, we identified, and the second case also we only identified. And we used to do sampling procedure, everything. Within 2 days, we used to
Yeah.
run all the samples.
Yeah. I mean, that's certainly a great thing, and you have been the first group, to have done the sequencing and uploaded all those kind of things, including the Omicron. But what amazes me is, Sunil, being a cytogeneticist, looking into this genetic sequence, the genome sequencing, and also looking into the lateral aspects. So, for example, you might be very busy, analyzing some panels, like fish panels or any other cytogenetics panel, all those particular panels, and still you would get time to do this much job, which is quite an amazing thing. Yeah.
Yeah, actually, during my PhD, we worked on hepatitis also.
Right. Yeah.
Hepatitis A, B, C, and E. So we worked on hepatitis. Because of that, a little bit helped me to look after the SARS-CoV genome sequencing. Because
Right.
during the time we used to do the hepatitis B genotyping and C and E
Right.
genotyping. So that helped me to do this sequencing little bit easy for me
Right.
to do the analysis part.
Right. Right. One of the other things which I always fond of asking you, Sunil, I know, there are certain diseases like, AML or ALL, Subbu is an expert in this, acute lymphoid leukemia and myeloid leukemia. There are certain, I think, fish panels and dish panels that are readily available, the fluorescence in situ hybridization, panels for
Yeah.
screening, ALL and also, for other things. I mean, although these have been in place, how sensitive are these fish panels for detecting such leukemia for early born or newborn children? Sunil, can you please highlight, especially for
Uh,
understanding the, or maybe detecting those recurrent chromosomal abnormalities,
the
in
fish
these.
panels are there for ALL, and that are uh, fish panels are there, ALL and CML cases also. So they're the targeted panels from abort
Okay.
systems.
Right.
So any vision, the probes, any vision probes that are
Right.
specific and that are very
Right. Right.
uh,
Right. I mean, although we are trying to use it, do you think it's that cost effective? Still, do you think that a normal Indian could really afford these particular tests, or how is it? I mean, just because yours in the government lab, maybe
no, we doesn't charge.
or you don't charge, being a government analysis.
It's a
Yeah.
free of cost. For all the services are free of cost only.
Okay. I think otherwise it costs about 10 to 15,000, I believe, right?
Yeah.
Yeah, I think it's pretty
Mm.
expensive. Yeah. So do we
We
have these fish probes or fish-based panels, for all the
no
diseases right now?
no no no. We are not. We are only looking for aneuploidy screening.
I see. I see.
Aneuploidy screening, and now we are looking for Wilson's and Angelman syndrome. We are going to
Right.
start for Wilson and Angelman syndrome and Dijot syndrome, 22q deletions.
Right.
So we are planning to.
And do you report them to your to those consortia, to those particular databases for newborn screening every time? Is there any?
Actually, our newborn screening data is being compared with. We will be submitting the quality control to CDC Atlanta.
Oh,
So
I
our
see.
results are validated, 100% validated with CDC Atlanta.
Oh, wonderful, wonderful, Sunil. Yeah, this is what was my question. So, maybe, yeah, wonderful, Sunil. I think, I mean, one thing, another thing, I really wanted to tell the audience about Sunil is, um, he's a psychogeneticist, but if you really talk to him on any other particular things, so for example, you talk to him on cancers, or you talk to him on, say, pediatric diseases, or even the vitamin K project, that I have, sometimes I discuss with him, or maybe, any other particular things. He has, probably a solution, and is wonderful, persona where, you always keep smiling, a very healthy smile, very, I would say, determined smile, I would say, and bringing those particular things, taking all the odds, and what amazes me is he also puts a healthy work-life balance. You know, that's how he has been leading the lab. He has been doing those particular sequencing efforts apart from being a cytogeneticist, and sincere congratulations and appreciation to the lab at Loknath Hospital that he has been leading over there. Wonderful, Sunil. Thank you so much for this wonderful time. I have another one question. I really don't want to start this conversation like this, too early. There is another thing I really wanted to ask, but any questions from the audience?
Yeah, Prash.
Yeah, Sudhu, please. Yeah.
Yeah, that was wonderful hearing you, Dr. Sunil. So nice to listen to you. Yeah.
Thank you, Sunil.
Uh, so yeah, regarding with respect to something based on the MRD, the minimal residual disease, we see a lot of extraordinary setups which has been there in German, then US, in developed countries. And with regard to MRD, the minimal residual cells, with regard to AML and ALL, and even we also look at multiple myeloma. Is there any panels that it is developed from Indian point, or we still use the panels what the Western uh, like the scientific fraternity what they use?
No, we are following the same pattern like Western people only. We are not having our panels. We have to plan. We have to think.
Okay,
We have to plan the things.
okay. How do you like what kind of like markers which are like if you are aware of that any specific panel which talks about specific to AML?
AML, I think we can plan. Actually, I'm not having that much idea about AML panels. I have not linked, but we have planned for some gene therapy related project with AML. So one of our hematologists from pediatric hematology professor has planned some gene therapy targeted panels for AML cases. So I can discuss and I can update you if anything they are having the panel list. So I can share with you.
Yeah, part of my PhD in postdoc is on AML and ALL.
Okay.
So, currently I'm working with the CMC Ludhiana and few hospitals, cancer hospitals,
Okay.
specific more to AML and ALL. So when I was talking to the doctors, they're saying that if a kind of panel that can be established from Indian context, it'll be really a good sign
No, no,
for
because we have to
especially people.
look about the prevalence and incidence of the cases. So then we can plan some panels. What is the incidence?
Okay, okay. Yeah, sure. Thank you so much. Yeah.
Uh,
Yeah, wonderful. Yeah. One final question, Sunil. What's in store for Sunil the next 10 years? Where do you see the next 10 years, with a wonderful, genome sequencing lab that you're associated with, doing the newborn screening and all those wonderful things? Where do you see the next 10 years?
Actually, I am planning some projects. So I submitted some projects to ICMR. So I'm looking to do some. This is what I'm saying. Expanded newborn screening. That is my next project proposal that is in the process. So if it comes, then it will be a very good study. So we can benefit a lot of children, and we
Right,
can give a new life to the children. That is the main target.
right.
So
Wonderful.
mm,
Wonderful. Yeah. Thank you. Thank you so much, Sunil. So friends, this is Sunil Kumar Polipalli, who is a senior scientist at Loknath Hospital, where we had a wonderful conversation with him. In fact, Sunil and his wife, both are wonderful scientists, and I have a wonderful, I would say, young scientist, a junior scientist in the form of Yuvraj Polipalli, who is my good friend, in fact, and he's also he has been from the first batch of bioinformatics for school children, and he also asks wonderful questions, and they have a wonderful scientific intellect going on in the house every time. Not only that, keeping his wonderful temperament, a kind of a cool temperament. Another thing that really fascinates me, fascinates me is the kind of the workaholism that everyone should really take. You know, so if someone probably laments at your scientific excellence, how do you receive it? If someone tries to bring some kind of incompetence to the core of competence. How do you react to that? These are the kind of things that one could learn from Sunil. I've learned a lot of things from him during my I would say 3 years of my interaction with him. And no doubt he's an exceptional scientist, and his group has been doing a kind of a yeoman service in newborn screening, and as well as you know various other diseases as well. Thank you so much, Sunil. I would request everyone to switch on your camera and have a wonderful photograph with Sunil. Please don't miss this opportunity. Thank you. Others, please. Yeah. Thank you. Thank you. Others, please. We'll just wait. Yeah. Okay. Yes. Thank you. Say three. I'll count three. 3, 2, 1. So bring up your wonderful 32 teeth outside. Yeah. Thank you. 3, 2, 1. Yes. Other one. 3, 2, 1. Thank you. I think Indrani is missing here. Oh, I'm sorry. She has a question. She should have been here. Anyways. Yeah. Yeah. Thank you. Thank you, everyone. Thank you so much, Sunil, for your wonderful celebration.
Thank you, Prash.
Excellent, excellent, Sunil. Thank you.
I'll send the recording, everyone. No? Thank you. Thank you so much. Thank you, Sunil. Thank you so much.
Thank you,
From
Prash.
my heart. Thank you.